Hi, everyone. Welcome to the complement system lecture. We first introduced complement during the immunology topic, and then it was listed as an immune phase reactant, and it was part of the innate immune system. It had to do with inflammation, and it also helped lyse bacteria and other cells. So why continue to talk about complement? Well, complement has had a significant impact in the lab. It used to be the way that HLA typing was done. When you had antibodies to HLA antigens, you could introduce complement and lyse cells to recognize the ones that had their antigen that corresponded to the antibody. Complement has other reasons for being worthy of study. So let's, if you take serum and you electrophoresis it, which means you put some serum in a gel and you run a current through it, it separates the parts of the serum by size and by charge. So you can see here, it's a little small, but the smaller molecules travel the fastest, and the ones with the largest charge. And the biggest peak in the biggest component of serum is albumin. Albumin is a protein that maintains the osmotic balance in blood. It keeps water from just escaping the blood vessels. And so complement is part of the beta globulin portion of serum. So there's alpha globulins, alpha 1 globulins, alpha 2, beta globulins, and gamma globulins. Antibodies are in the gamma globulin region of serum, and complement is in the beta globulin region. So complement are proteins that participate in the innate immune system to enhance the ability of antibodies and phagocytic cells to clear microbes and also damage cells from an organism. Complement promotes inflammation, and they attack a pathogen's cell membrane and cause it to lyse, to lose its integrity as a cell. The activation of complement can be initiated via 3 distinct pathways. There's a classical pathway, a lectin pathway, and an alternative pathway. Each of them end up with creating a membrane attack complex that is responsible for lysing cells. So this depicts the roles of complement. It opsonizes cells, and so basically coats them to make them more recognizable for disposal by the intravascular phagocytic system. It can punch a hole in the cell membrane and hemolyze and lyse cells. Chemotaxis is it attracts cells to the area. Anaphylatoxin activity is another way of saying that it causes inflammation, and it also is responsible in activating cells by providing a signal, a stimulation to them. The disposal system's role of complement has to do with clearing older cells from the circulation by also opsonizing them. And then it also interacts with cells in the adaptive immune system through chemotaxis. And all these other processes here. I'm not going to go into those. They're probably not going to be tested on them, but even though it's not part of the adaptive immune system, it does interact with cells of the adaptive immune system. So the methods of destruction and removal of foreign antigens is through direct lysis via that membrane attack complex. membrane attack complex is a structure comprised of all these different proteins, C5a, C6, C7, C8, and C9. They form a circular structure on the cell membrane, and that causes intracellular material to escape and extracellular material to enter the cell, causing lysis. Coating or opsonization of foreign cells with C3 initiates phagocytosis. It makes them more recognizable for phagocytic cells to destroy them. And it also recruits cells for the adaptive immune system to the site of infection. These are the molecules that have the characteristics, specific characteristics. So C3A, C4A, and C5A are anaphylotoxins. That means that they participate in inflammation, and they cause a release of histamine from mast cells and other cells. C5A is also a chemotactic. It means it attracts neutrophils, basophils, mast cells, and monocytes. And finally, C3b is a powerful opsonin. It means it coats cells so that the intravascular or basically any phagocytic cell can remove them, recognize them and remove them. So how is the cascades are have an initiation point, and it causes molecules to cleave, and then they have an active moiety that leads, they're enzymes that lead to other reactions. And typically cascades have both activation and downregulation properties associated with them. At the end of this presentation, you'll see a video that will explain in detail how some of these mechanisms work. Right now, I'm just introducing the high-level concepts about complement. So the classical activation pathway begins with C1. C1 circulates in the plasma as an inactive complex. And when it encounters one IgM molecule that's bound to a membrane, so it encounters the Fc portion of the IgM, or several IgG molecules in close proximity. So it interacts with the heavy chain, the Fc portion of the two adjacent IgG molecules. Then it activates C1q, and together with C1r, C1s, and C1s, the C1 complex becomes an enzyme that activates the next step of the cascade. A second way of activation is the lectin pathway. The lectin pathway is triggered by carbohydrates in microbial cell walls, so it doesn't require immunoglobulins. So whenever this carbohydrate is in the blood, it can activate C1. And then not all immunoglobulin molecules are capable of activating complement. The most efficient one is IgM, being a pentamer and a fairly large molecule. And then there are subclasses. 3 of the 5 IgG subclasses are capable of activating complement, and those are IgG1, IgG2, and IgG3. The alternative activation pathway is mediated via properdin. Properdin B is released from activated neutrophils and is bound to the cell membrane of bacteria and dying human cells, where it recruits, stabilizes C3b to form alternative pathway C3 convertases that lead to the formation of the membrane attack complex. So properdin is a here real quick. It's a glycoprotein that is secreted by the white cells for the purpose that I described. So the alternate pathway is normally antibody independent, but it can be initiated by IgA and IgE. So the classical pathway is initiated by IgM, IgG1, IgG2, and IgG3. The alternate pathway typically is antibody independent, but it can be initiated by IgA and IgE. And then I mentioned earlier how there is activation of the cascade, and there has to be some downregulation of the cascade, otherwise it go on forever until some bad things happen. So factor H is a member of the complement system that downregulates complement activation by preventing C3 conversion to the active form of the protein. So now let's take a look at that video. And I hope you enjoy it.
Plays a major role in the killing and clearance of invading pathogens, including bacteria, viruses, fungi, and parasites. The system is composed of over 30 proteins, most of which are found in serum, while the remaining proteins are membrane-bound proteins and receptors. On activation, complement proteins that participate in the activation of either the classical or alternative pathways of complement interact in a highly specific enzymatic cascade and generate proteolytic fragments that mediate the numerous biological functions of the complement system. In this video, we will focus on the activation and biological functions of complement. Activation of the classical pathway. The classical pathway is typically activated by antigen-antibody complexes. Although there are many other activators of this pathway, only two isotypes of antibody, IgM and, as shown here, IgG, will activate the classical pathway. While one pentameric IgM antibody will activate the classical pathway, a minimum of two IgG antibodies in close proximity are required. In either case, antibody must be bound to antigen in order to activate the classical pathway. The classical pathway is activated when C1, the first protein in the pathway, binds to the Fc portion of the antibody. C1 is a large macromolecule composed of a C1q molecule and two molecules each of C1r and C1s. It's the globular head portion of the C1q that binds to the Fc binding site on the antibody. This binding causes a conformational change in C1q and subsequent autocatalytic conversion of C1r to an active serine protease. Activated C1r cleaves C1s to an active serine protease. Activation of C1 by binding antibody is also termed complement fixation. The next step in the activation of the classical pathway is the cleavage of C4 by the activated C1 molecule. C4 is cleaved by the activated C1s molecule in C1 into two proteolytic fragments, a small peptide called C4a and a larger fragment, C4b. C4b attaches covalently to the antigen-antibody complex and has a binding site for C2, the next protein cleaved in the classical pathway. C2 is also cleaved into two fragments, and the resulting C2a fragment binds to C4b. This bimolecular complex of C4b2a is a C3 convertase of the classical pathway and serves to cleave C3 through the enzymatic activity in the C2a portion of the complex. Classical pathway C3 convertase. The C3 molecule is cleaved by the C3 convertase into C3a and C3b. The C3a fragment has potent biological activities, which will be discussed shortly. The C3b fragment can covalently attach to the C3 convertase, resulting in a trimolecular complex consisting of C4b, 2a, 3b. This multimolecular complex is called the C5 convertase of the classical pathway and is specific for cleaving C5. Classical pathway C5 convertase. The C5 molecule is cleaved by the C5 convertase into C5a and C5b, both of which mediate important host defense functions of the complement system. Both C3a and C5a are potent chemoattractants. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. The terminal complement pathway, or formation of the membrane attack complex. One of the best-known host defense functions of the complement system is its ability to lyse many bacteria, envelope viruses, and nucleated cells. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis. Activation of the alternative pathway. The alternative pathway of complement is activated by LPS and other lipopolysaccharides found on the surface of invading pathogens. The alternative pathway is always active at a very low level. This activation is due to the spontaneous hydrolysis of an internal thiol ester bond in C3. Hydrolyzed C3, called C3 water, has a binding site for factor B, another protein in the alternative pathway. Factor B binds to C3 water and becomes a substrate for cleavage by factor D, denoted here as FD. Factor D cleaves factor B into two fragments, BA and BB. The enzymatically active BB fragment remains associated with C3 water, and this bimolecular complex is the alternative pathway C3 initiation convertase. Alternative pathway C3 initiation convertase. This initiation convertase cleaves C3 into two fragments, C3a and C3b. The newly generated C3b can covalently attach to nearby surfaces, just as C4b does in the classical pathway. In the absence of an appropriate activator for the alternative pathway, complement regulatory proteins block further activation of the pathway. However, C3b, on interacting with an activating surface, such as a bacterial cell, will bind factor B, which will be cleaved by factor D, resulting in the formation of the alternative pathway C3 convertase of C3b-BB. Alternative pathway C3 convertase. The C3 convertase of the alternative pathway cleaves C3 to C3a and C3b. Some of the C3b generated by this cleavage does not interact with the activating surface and remains soluble. Some of the C3b generated will covalently interact with the activating surface and start the formation of additional alternative pathway convertases, thus rapidly amplifying activation of complement through this pathway. The surface of an activator, such as a bacterial cell, can rapidly be covered with many thousands of C3b molecules in a few minutes as a result of this amplifying effect. And, as with the classical pathway, some of the C3b generated will interact with the C3 convertase itself, resulting in the formation of a trimolecular complex of two C3b molecules and BB. This multimolecular complex is called the C5 convertase of the alternative pathway and is specific for cleaving C5. The C3a and C5a fragments generated by activation of the alternative pathway mediate the same functions of chemoattracting and activating granulocytes and phagocytic cells. C3a attracts mast cells to sites of complement activation, and binding of C3a and C5a to these cells, as well as basophils, induces degranulation and release of histamine and other vasoactive amines. C5a is a chemoattractant for macrophages and neutrophils, and binding of C5a to these cells primes them for mediating their host defense functions. Activation of the alternative pathway results in the formation of a membrane attack complex, leading to the lysis of the invading pathogen. Lysis of cells by the complement system is mediated by a large macromolecular structure called the membrane attack complex, or MAC. The MAC is formed through what is called the terminal complement pathway, which starts with the generation of C5b. C5b associates with C6, C7, and C8, forming a large multimolecular complex that associates with and begins to disrupt the cell membrane. Subsequently, C9 binds to C8, and this is followed by the binding of many additional C9 molecules, leading to the formation of a large pore in the cell membrane. The pore in the membrane is large enough to allow water, ions, and small molecules to enter the cell, leading to lysis.
Now, there was a lot of detail in that video. To get a better sense for the most salient details, the most important ones, I recommend you read the technical manual section on complement.
Yes.
Sorry about that. Let me stop that.
Unbelievable thing that could happen.
English. I already got it.
And then I also want to mention that when you get to autoimmunity, cold autoantibodies, warm autoantibodies, complement, what you've learned about complement will become pertinent again, because these cells will be coated with complement molecules, and there will be some lysis associated with that involvement. I know this was a lot of information. Please take a look at what's in the technical manual to help you prioritize the most important salient points. And with that, this is the completion of this lecture. Thank you for your attention.